Welcome to your General Botany Virtual Lab on shoot growth and development. We're going to talk about how a tree trunk this big developed from a tiny stem. Okay, so welcome to today's lab. Similar to last week's lab, we are going to be talking about the growth and development of plants, but this time instead of roots, we'll be talking about shoots. Remember, roots are the kind of below ground structures that function in absorption of water and nutrients, while shoots are the stems and leaves. These are usually the above ground structures. This is just some generic plant, let's say, and this is sort of its young development as a seedling. We call this primary growth because it is elongating, and this is the first thing to happen in a plant. It needs to grow up because that is where the sun rays are. So this is the most important thing, and it's the very first thing to happen. And we call this primary growth. But plants aren't just extremely tall and skinny, narrow like that. No, they also grow in width. And just like we learned last week, how roots can become woody. And we can kind of see that here. Well, stems can do the exact same thing. There's probably not news to you there. But what's going on in this picture is a tree has started growing on top of a stump, and it's got some woody roots that we can see below, and its woody stem is up above. And it is also on a big dead woody stem. So yes, they grow in both length, primary growth, and width, which is secondary growth. We're going to talk about both of those in today's lab. Primary growth happens at all of the root apical meristems and all of the shoot apical meristems at the same time. It's not just happening from a seedling. Primary growth, it kind of sounds like it's just the first growth, and it is. It includes that very first growth. But as you can see highlighted in red on this image, primary growth is happening in many, many places on the plant simultaneously. Secondary growth can only happen somewhere where primary growth has happened. First primary growth and then secondary growth. But they're happening at the same time on an individual. So in this woody tree, we have secondary growth in all of these places where you can see wood, and that's increasing, of course, the width. And at all the tips of the branches where we see young green buds, that's where primary growth is also happening at the exact same time on the same individual. Same story below ground. But we're focusing today on the shoot growth and development, again, the stems and the leaves. Not all plants can do secondary growth. This picture on the left is a cross-section through a stem of a plant similar to this one. This is some lily relative. Lilies and grasses and aloe and even things like palm trees, these are all in the group of plants called monocots, which we're going to talk about later in the semester. Just know that monocots, they're not able to go through secondary growth. They're only able to do primary growth. And part of the reason is the organization of their vascular bundle. Notice how the vascular bundles are sort of arranged in a haphazard fashion around the interior of the stem. So imagine that this cross-section was kind of taken right through the stem. This is opposed to the way that eudicots have their vascular bundles arranged. Nice and neatly organized around the outside of the stem, all the way around the perimeter of the stem. The organization of this, as you'll see here in a couple of minutes, allows for some pretty interesting types of growth. So again, primary growth is the elongation of plant parts. Let's zoom in on one of these young plants. So what we did to get from that previous picture to this one is I took a longitudinal section. This is the very, very tip of that growing stem. Notice how we've got this area of pretty dark cells. They're kind of dark because they're very small, and they're very small because they're brand new. And that is a sign that we are looking at the shoot not the root, the shoot apical meristem. All right. We've got a few other tissues that we can label on this. Immediately after the shoot apical meristem starts dividing and producing cells, what it does is it produces essentially 3 layers. Let's start with the innermost layer. So everything I'm labeling here in green is ground meristem. It's all of this sort of light blue parenchymus looking tissue. The next layer that's visible is this sort of line of tissue right here, and this is called the procambium. The third meristematic layer derived from the shoot apical meristem is the protoderm. Now in this picture we've zoomed out a little bit, so we had just been focused right there on that small, very tip, and now we've still kind of got those same labels. We need to continue talking about sort of the older part of the stem, so what I'm going to do is show you sort of a cross section. Again, if we were to take like a fine razor and just slice this part of the stem out, and I'm going to show you what that's going to look like as it develops to be about this old. So we're going to look at this time increment now. Through the young shoot apical meristem shows exactly 3 meristematic layers that are derived from the shoot apical meristem. Those are the ground meristem, procambium, and protoderm. Over time, those 3 layers will continue to develop. The protoderm will become the epidermis, the ground meristem will become both pith and cortex, and the procambium will become primary phloem and primary xylem. And here's what that might really look like if you were looking at a stem under the microscope. So these are the vascular bundles. There are a whole lot of them. That means that this part in the middle must be the pith. And I wanted to tell you that there's a couple different sort of regions in the vascular tissue area and in between the vascular tissue areas. So there are these terms fascicular and interfascicular regions. And that word fascicle, which is the root of fascicular, means bundle. And it's referring to these vascular bundles here. So if we were to describe the 2 regions, we would say these regions, sort of within the bundles, are the fascicular regions of the young stem, while these regions in between each one of these vascular bundles, these are the interfascicular regions. Where we left off in our last cross section was about right here, pretty young part of the stem, remember? Now we're going to kind of watch the development till, just about here or so. This is where we had left off on the diagram. When we look closely into the vascular bundles, there are actually some cells in between the xylem and the phloem that are called undifferentiated procambium. It's that undifferentiated procambium within the vascular bundles that's going to become the vascular cambium. The vascular cambium is also derived from the regions in between the vascular bundles, so that's the interfascicular cambium, while the fascicular cambium is just the cambium that's derived from the vascular bundles. Now, this is a demonstration of how the vascular cambium will divide and differentiate cells both to the inside and to the outside. The cells also have to divide laterally in order to account for the increasing diameter of the vascular cambium. Here is what this would look like in real life if we had a fine cross-section looking at this stem under a microscope. So let's say we were to zoom in on, say, this vascular bundle right here. That's what we've done. what we notice is that there is sort of this line of cells that are much skinnier. They're sort of like these pancake cells. You see those? They're sort of pancakes stacked on top of one another even. And the reason why they're so flat and stacked is because they just divided. These are your meristematic cells of the vascular cambium that has just formed at this stage. So what I've just labeled in red here and was labeled in red sort of right here, this is the fascicular cambium. So this is the part of the vascular cambium that is formed within those vascular bundles, and that's why we call it the fascicular cambium. The other type of cambium here that sort of connects the fascicular cambia to one another is the interfascicular cambium, and this is sort of labeled in green here and on this little diagram as well. So together, the vascular cambium ends up being one line through the interfascicular cambium and the fascicular cambium. And we can see that by doing this. Right on the outside of the xylem and the inside of the phloem, continuously through that interfascicular region and then into the next vascular bundle, we get this continuous line of vascular cambium all the way around. This is what is so important, the fact that the vascular bundles make a full ring. Now, our meristematic cells that are part of this vascular cambium can start dividing. And that's what we're seeing here. The vascular cambium cells have divided, and they've even differentiated now. The red cells to the inside represent that first layer of secondary xylem, while the blue on the outside of the secondary phloem. Now remember, sometimes these cells need to divide. These vascular cambium cells will divide laterally just to increase their own perimeter, since the circle is expanding, since secondary xylem is accumulating. So here's what that's going to look like as the vascular cambium starts to divide rapidly and accumulate cells that are differentiating to the inside and to the outside. Our vascular cambium is still in black here. The dark red is the new secondary xylem, while this dark blue is the new secondary phloem. The primary phloem and the cortex and the epidermis, they're just getting pushed out from the accumulation of those new secondary tissues. So now we're zoomed in on a slightly older cross-section of a stem. But again, we're looking through the microscope at this image, and this is a very, very thin slice that has been dyed for us. So these kind of pinker cells down here, these are all the xylem. This then, right here, this must be our newly formed vascular cambium. So now we know with the vascular cambium, as it's dividing, everything to the inside is xylem. So all of this here, this is all secondary xylem, except for maybe like these little clusters of xylem cells down here. These might be primary xylem, but it's kind of hard to tell at this stage. The majority of all of these cells here are, and that means that everything up here must be secondary phloem, because those vascular cambium cells divide to the outside and they produce that. Okay, so now we need to account for those layers that were outside of that primary and secondary phloem, which, by the way, maybe there's some little chunks of primary phloem sort of scattered throughout, but they would have been pushed out by the secondary phloem. So all of this up here, this must be our cortex. Remember that has been getting pushed out. And same with the epidermis. The epidermis is also getting totally pushed out. Wanted to just really hone in on this vascular cambium. So now what we're going to do is take a slice of these cells and we're going to zoom in on it and see exactly what that vascular cambium is doing. So let's just take like this slice of cells right here.
And now, so we've got our vascular cambium, we've got our secondary phloem, and we've got our secondary xylem. And here's what that would look like sort of in a diagrammatic view of cells dividing and expanding. So in this picture, our vascular cambium, which I've been drawing in black, is now this yellow line. And notice that it is sort of expanding out. The yellow line here, the vascular cambium, starts sort of on the interior, but as it's accumulating secondary xylem to the inside, that vascular cambium is inherently itself being pushed out. And that is sort of what we're seeing in this diagram. So every time one of these cells divides, it is producing this secondary xylem to the in. And then as it gets older, again, these cells will divide and they will continue producing cells to the inside. So here, on this kind of oldest version of our little strip of cells here, the oldest xylem that was produced earlier is back here. That means that this would be the youngest xylem. This is brand new xylem that was just formed from that vascular cambium. And then it's the same story, but to the outside with the phloem. The phloem is being added right here, right next to the vascular cambium.
Go back to our drawing diagram and see what this looks like. As secondary growth continues, secondary xylem accumulates and pushes the vascular cambium and everything else to the outside. Secondary phloem is also accumulating, and that's pushing the cortex and epidermis to the outside.
Now, here is another cross-section of a stem that's a bit older, but we can still see all of our important parts. Now, this right here that I've just drawn in black, but I'm going to draw again in white because to make it more visible, this is the vascular cambium here. And that means that all of this stuff to the inside, yes, all of this is all secondary xylem. And at one point, this vascular cambium was on a more narrow part of the stem. Like when the stem was younger, it was right here only. And then only this would have been the secondary xylem. But that vascular cambium is being pushed out and out and out as it is continuing to grow with age. Okay, and then we need to account, again, for the stuff that's going on the outside. This is all cortex out here, and we've got something kind of exciting going on out there. As our cortex and our epidermis is being pushed to the out, bark is going to be forming, which we can sort of start to see here in this picture. This is all going to be bark on the outside. So this is a picture of a much, much, much older stem. Previous picture we were just looking at, this was maybe only 2 or 3 or 4 years old. This stem, though, is many, many years old. And we know that because of all of these lines. Okay, so the truth about wood is that all of this woody, hard stuff on the inside of a stem, it's all secondary xylem. It's all produced from the vascular cambium. So let's look back at this picture. Now, this is a slice from our picture we were looking at earlier. We had our vascular bundles, and remember, the cortex was outside of these vascular bundles. The cortex is derived from the ground meristem. Remember, the ground meristem, it produces pith to the inside of the vascular bundles, and then all of this on the outside of those vascular bundles is cortex. And as our vascular cambium, which has formed right here, is pushing cells to the out, here's our vascular cambium here, and it's pushing cells to the out, that cortex is being pushed out. The epidermis is being pushed out. And now our cortex is approximately this area right here. And we've kind of got most of these other terms that we're already familiar with, except for this new one right here. Periderm. Periderm is a new term. This is a new layer of multiple types of cells, actually. But the periderm is derived from the cortex. So the periderms are actually coming from another cambium. What? Yes, we've been talking about the vascular cambium this whole time so far. And that would be represented by this line in this sort of fancy cutout view of a diagram of a piece of wood. And that means, again, everything inside here is secondary xylem. Well, our cortex was pushed out, and it formed this new layer of cells called cork cambium. It's outside of the vascular cambium. Just like the vascular cambium, that word cambium that's in cork cambium, it just implies that we've got a layer of meristematic cells that are continuously dividing. And just like vascular cambium, the cork cambium, too, is dividing both to the in and to the out. And it's got two sort of specialized types of cells. Ones that form to the in and ones that form to the out. We're going to come back to this slide a little bit later to talk about those inner and outer barks.
Going back to our diagram drawing one last time, a new layer of cells is formed from the cortex called the cork cambium. Since it's the cambium, we know it's going to be meristematic. It divides cells to the inside. These are called phloem. And as it's dividing, it also differentiates cells to the outside. These darker brown ones on the outside are called cork. And they're produced to the outside of the cork cambium. Now, if we zoom in on a little section of the cork cambium, and we look at the first layer of periderm. Here, we've got cork cambium, and this is showing how they divide and grow and differentiate. And the cork cambium will always be present. It'll always be that thinnest layer of cells. Once they do start differentiating, those lighter tan cells to the inside are called phloem. Again, the cork cambium, it'll continue dividing. It'll remain there, but the cells that have differentiated to the outside have become cork in dark brown. The word periderm is literally another word for outer bark. And if it were a math equation, a periderm would equal phloem plus cork cambium plus cork.
So this periderm that's up here, this is actually, if we zoom in on that, it's actually 3 layers. So let's look at that. If we zoom all the way in, now this is probably a different individual stem than this one, but it's got the exact same idea. The cortex is sort of this area down here. It's labeled as collenchyma and parenchyma, and that makes sense. Usually cortex is made of mostly parenchyma and collenchyma cells. And so together, you would say these are the cortex. The very outside of the cortex here, though, it has started to differentiate. And what we're seeing is this one very, very, very thin layer of cells, and this would be the cork cambium. So it's going to be approximately right here. It's literally just one cell layer thick. That gets a little crazy around there. It looks like there's some actual cell divisions taking place in this image. This is our cork cambium. And what it's doing is it's producing phloem to the inside. And so these cells here are the phloem cells. These are phloem. And then it's also producing some cells to the outside. It's producing these cork cells to the out. So cork cambium produces cork cells, and it produces phloem cells. Okay, now we're looking at a cell that has been producing cork cambium and cork cambium products for some time now. And we've got a couple of pieces of evidence of that. Again, we've got our cortex just on the inside. Again, we're just zoomed in on a very outside part of a developing stem. And this layer of really thin cells, approximately right here, they really do kind of look like pancakes stacked on top of one another. This is our cork cambium. And that means everything to the outside here, this is all cork. And in fact, what happens is we might even end up with multiple layers of cork cambium cells, and therefore multiple layers of entire periderms. So as that cork cambium is producing that cork to the outside, this must all be phloem then. But I think what's happening in this picture is the cork cambium, it's produced from different cortex layers. And so a cortex cell can differentiate into a cork cambium, and it sort of appears that that is what has happened right here too. There's almost this other layer of cork cambium that we're seeing right here. And it will always be doing this. It's going to always produce cork to the outside. So these are both producing cork to the outside, and then it'll always be producing phloem to the inside. And so you might end up with these layers of periderm. So now let me define this word periderm for you, which I've been using many times without defining. A periderm is all three of these layers together. So in other words, the periderm is the cork cambium plus its cork plus its phloem. And then when I say periderms, I'm talking about potentially multiple layers of cork cambium and its products. Now here's an even older one. Again, we definitely have a couple of cambia in this picture. Actually, right here at the very bottom, this must be the vascular cambium, because I'm seeing some vessel cells here. These vessel elements must be xylem, and everything to the outside here, this looks like phloem. So we've got that vascular cambium down there. We're seeing potential cork cambium. And again, there might be multiple layers of cork cambium up here too. So this must all be phloem to the in, and then this must all be cork to the out. Now, the epidermis is just being sloshed off. I haven't even really mentioned it much. It would just be this very outermost layer. Maybe there's a cuticle on top of that. That's kind of what this white stuff is, I think. Yeah, the epidermis is no longer needed. It was just one cell layer thick, and the periderms are much more effective at keeping pathogens out. Because remember, as this stem is expanding, it was at one point a very small circle, and it's getting bigger and bigger. And so the outsides, the perimeter of the outside is getting bigger. In which case, it must be sort of cracking. In order to expand, when it cracks, it could be potentially letting pathogens in. And so this cork cambium and its cork and phloem products are extremely important for the plant to prevent disease. So now we're looking at a stem that has gone through approximately 2 years of growth. This right here must all be the secondary xylem, but it's sort of split into 2 years. This would be the first year's worth of secondary xylem. Maybe the primary xylem would be these little sort of bundles that we can barely see anymore at this point. This would all be the second year's worth of secondary xylem. And that means that this line right here, this must be the vascular cambium right here. And then we've got our cork cambia kind of on the outside here, producing the cork and the phloem. Here's a different stem that's in its third year of growth now. Just to show you how those layers of xylem will look year after year. They sort of end up looking like the xylem layers are sort of stacked on top of one another. You see that? Okay. Well, there's something very useful about the way that those years of secondary xylem stack on one another. They continue stacking year after year. And when we look at a very old stem like this, that we know now is just entirely made up of secondary xylem, we can now interpret what these lines mean. So let's take a closer look at this. Let's zoom in on this part of the stem right here. So that's what we've done in this picture. Now we can see a little bit more closely that we have this very consistent pattern of lines on top of one another, line after line. These are all different years of secondary xylem. Let's look a little closer. Let's zoom in, say, on this section right here. Now, this is a very, very thin cross-section that we're looking at under a microscope. We can tell because the light is sort of coming from underneath. We can see the light just very clearly through these large vessel elements from the xylem. Now, these are not cambium lines. No, these are yearly growth increments. And they do sort of form this pretty distinctive line. Let's look even a little bit closer. Let's zoom in a bit more here. What happens is that during the, at least up here in the temperate zone of Earth, what happens is each new year's growth starts very wet, right? The rainy season is early spring, and so it's going to produce these large vessel elements early in the season. It's got lots of energy, lots of water, lots of nutrients available to it because it's rainy season. And so up here it's producing these larger, more conductive cells. Same thing with the next year. It was early in the year, it was producing these larger vessel elements. But as we get sort of later in the year, so let's go back to this one, this year increment right here. This might be later in the year, after the rainy season. It's no longer producing those larger vessel elements. Same thing with up here, that next year. It would only produce those large vessel elements for the rainy season, essentially. During the drier season, throughout the dry summer, it's only producing smaller diameter vessel elements or tracheids. So let's actually zoom in just a little bit more, like right there-ish. Now, in this picture, we can see a couple of things. The early wood vessel is what we're going to call these really large ones. And then these late wood vessels are the smaller ones. Again, the early wood has more access to water, and so they're able to produce larger, slightly more efficient vessel elements. Okay, so zooming back out a little bit, we see that every single year there's a wet season and then there's a dry season. And the wet part of the year will result in the xylem sort of looking like it's a bit lighter, like we're sort of seeing there. It lines right here that represent sort of the drier season, where the cells are much smaller and therefore they appear darker. Now this next picture, this is on the right here, this is a scanning electron microscopy image of a chunk of wood that has been sliced very nicely and neatly with a razor. This is a picture that's been zoomed in many hundreds of times, and it's not a normal microscope. This is a scanning electron microscope. And what I've done here is I've sort of aligned this picture so that it's sort of going with the angle that we're seeing here, because this right here is a two-dimensional version of the top of this three-dimensional cube of wood. And so these lines right here, that line, those correspond with, this line right here and this line right here, because we've got those larger early wood vessels kind of right by those lines in both of these yearly growth increments. And then when we look at the side view, so this is a two-dimensional version of the side view of, say, something kind of like right here-ish. And we can see a few things. First of all, we're now seeing a longitudinal version of these vessels. And then there's sort of another one. We're just essentially literally seeing the side view of one of these, and this would be that top view like that. And then this would be that side view of that same vessel element like that, okay? There's something else that we can view now. Now that we're seeing sort of this whole composite of many years of woody growth. There are obviously longitudinal vessel elements. They are bringing water from down below ground to up above ground, and so they are vertically held. But there are these other cells that are growing laterally. They're growing out to the side, sort of the opposite direction, or perpendicular to those vertically held, those vertically stacked vessel elements. These lateral cells are called rays. And these are parenchyma cells that are helping move nutrients to and from the secondary phloem and the secondary xylem. They're moving products from the phloem over to the developing xylem where it's needed. And most importantly, it's moving water that's being absorbed and taken up from the roots out to the phloem, out to the cambia, and out to all of those other outer cells, all of those other meristematic cells that are in dire need of that water. And here's just a closer look at that. So here is a real picture of a chunk of wood where we've cut out a slice just so that we can really see inside of this wood. And so all the things we've been talking about are present. We've got our vascular cambium way out here on the outside. And that means that everything to the inside here, this is all true wood, which is secondary xylem, and that means everything to the outside right here, this layer just outside of that vascular cambium, this is all secondary phloem. And that means that everything to the outside of the secondary phloem, these are our periderm layers. So back to what we were just talking about a minute ago, it's these lateral sort of radiating lines of parenchyma cells that we call rays. And when they're within the xylem, they're called xylem rays. And then they do actually stretch out to the phloem, and out there we would tend to call them, well, phloem rays. We could also potentially see some of those rays kind of in the side here. It's a little bit harder to see, They may not continue sort of all the way down. Maybe they're just sort of strips of these rays, sort of like this, stacked on top of one another. Maybe there'd be another one like right here or something. Cool, these are all rays. They're really moving water around the width of the stem at this stage. Now, we've already talked about what these individual dark lines, or these lighter and darker lines sort of represent. These represent yearly growth rings. And the reason why they're darker than light and then darker than light is because of the seasonality here, right? We talked about that earlier. But there's another sort of dark and light section that we see within the secondary xylem. We've got this whole area right here that's much darker than this area of wood to the outside. It's only this part that I've just labeled in magenta on the outside. This is the only actual conductive secondary xylem anymore. Yeah, this is what we call the sapwood. It is actively moving nutrients and water up the plant. Everything here on the inside, all of this darker wood, it's dead. Now, I know what you're thinking. Secondary xylem, they're sclerenchyma cells, and so secondary xylem is dead. It's true. Everything here that I've just circled in magenta and black, they're all dead cells, technically. The vast majority of these cells are dead because they're sclerenchyma. But what I mean by dead for this interior part is it is no longer functioning in moving water up the plant. We call this heartwood. Now, just because it's not moving water up doesn't mean it's totally useless and it's just going to rot away or something. It might rot away, but that'd be for a different reason. It is actually important for the plant. As it's growing in girth, it's going to need more support, more structural support. And this heartwood will remain in the center of the tree for as long as it grows. This hardwood will kind of continue expanding out, and it will provide more and more structure for the plant as it's growing older. Everything to the outside of the vascular cambium is secondary phloem. So everything that I'm sort of labeling here, outlining in blue, this darker area, this is all secondary phloem. And actually, everything to the outside of the vascular cambium, including this phloem, is part of the bark. The secondary phloem, though, is a specific part of the bark. It's called the inner bark. So really, what I just labeled in blue is right here. It's the layer outside of the vascular cambium, but inside of that cork cambium. This is all the secondary phloem, and we call this the inner bark. The outer bark is everything that's outside of the secondary phloem. It includes the cork cambium, and the cork, which would be on the very outside, and the Periderm, which would be in between the secondary phloem and the cork cambium. So let's look a little bit closer at that. Now, in this picture, what I've just done is we've zoomed in on just a little bit of, say, the outside right here. Okay. We know the vascular cambium must be right here because we've got our yearly growth increments of our secondary xylem below that. And then that means that everything outside of that vascular cambium, directly outside of that vascular cambium, must be phloem, which botanically we also call inner bark, namely secondary phloem, to be clear. The outer bark are all of the multiple periderm layers, and they're sort of labeled by these arrows here. Each one of these is a different periderm layer, but together all of those periderms, part of the outer bark. All of the periderms combined are outer bark. The periderms are expressed morphologically in so many various ways. Depending on which species of plant we're looking at, the outer layers of bark might be very smooth, they might be very chunky and produce a ton of cork, they might be sort of papery and peely, and it turns out that botanists use the look of bark, the look of that outer bark, to differentiate between taxa within the plants. At least sometimes. You, of course, can use other traits like roots and stems, but that outer bark really does come in handy because it's usually quite visible. So now, as we wrap up our virtual lab on shoot growth and development, we've talked about primary growth, secondary growth, we've talked about vascular cambia, cork cambia, and both of their products. I wanted to show you this picture. This is this old apparently old, gnarled tree that I found over on Mount Shasta a couple of years ago. All of these, they didn't even have their tops anymore. They had totally broken off, but I had imagined maybe at one point they were alive. Yet, these live green branches that you're also seeing in this picture, these are coming right off of the apparently dead part of this tree. So I have a question for you about this tree. How old is it? It looks extremely old. And so should we say this tree is hundreds of years old? Well, sure. It is hundreds of years old. Maybe here in the really old part of the tree, if we were to look at a cross-section way down there. But up here, and up here, this tree is only a year or less old. In fact, at these very tips of each one of these branches, of which there are a lot in this picture, they're just a day old or so, because the shoot apical meristems are present in all of the growing tips of every tree, even the oldest of the trees. And so, thinking back to that original video that I showed you at the beginning of this virtual lab of the old redwood tree, that tree was hundreds, possibly thousands of years old at its base. But there are hundreds and thousands, and probably more like hundreds of thousands of shoot apical meristems growing off of that old redwood, off of this old white bark pine tree. And so it is simultaneously hundreds of years old and just a single day old, depending on which part of the tree we're talking about. And I love that about trees. Okay, so this was the end of our virtual lab. Like always, I'd like to see if there are any questions or comments. Anyone? No? Anybody? Would anybody like to share anything with the class? Oh, it looks like we actually do have someone in the back there. Yeah, let me just change the screen here. It looks like our fellow student Holmes is here to demonstrate the rigidity and durability of secondary growth in woody stems. Notice how his very powerful jaws are barely able to break through that hard secondary growth produced from the vascular cambium. Also, notice the grassy plants all around him. Those are not able to produce wood. They've just gone through primary growth, not secondary, like this very hard stem that he is able to crush with his teeth. Thank you so much for sharing that with us, Holmes. We really appreciate it.